DNA is made up of nucleotides, each of which has a phosphate group, deoxyribose sugar, and a nitrogenous base. The sugar is bound on one side to a phosphate group and bound on the other side by a nitrogenous base. The four bases are adenine, guanine, cytosine, and thymine. Adenine always binds with thymine, and guanine always binds with cytosine. Two of the bases, adenine and guanine, are purines, double ring structures. The other two, thymine and cytosine, are pyrimidines, single ring structures. DNA contains two strands, which makes up the double helix. The hydrogen bonds between the phosphates causes the DNA to twist. The two strands of DNA are anti-parallel. Both strands run in opposite directions. One strand runs in the 3' to 5' direction, while the other runs in the 5' to 3' direction. Thymine and adenine have 2 hydrogen bonds, while cytosine and guanine have 3 hydrogen bonds. Moving on to DNA replication, the DNA molecule is untwisted by 2 enzymes, helicase and DNA gyrase. Helicase unwinds the parental strands, and DNA gyrase relieves the strain by cutting, winding, and rejoining the DNA strands. DNA unzips in an area called a replication fork. The formation of new DNA is initiated by RNA polymerase and carried out by an enzyme called DNA polymerase and a series of proteins. These cause the 2 strands to break apart. Nucleotides can only be added in the 5' to 3' direction. However, because of DNA's anti-parallel nature, one strand of DNA runs in the 5' to 3' direction, while the other runs in the 3' to 5' direction. Therefore, on the leading strand, which is 5' to 3', nucleotides are assembled normally, while on the lagging strand, 3' to 5', Okazaki fragments are created. These fragments are later joined to create one long strand by DNA ligase. Let's rewind a bit so we can talk about RNA primers. RNA primers are several strands of RNA added to the lagging strand of DNA by RNA polymerase. It will begin synthesis of the lagging strand in DNA replication. Primers start the synthesis of the leading strand, and each DNA fragment of the lagging strand. DNA replication results in 2 semi-conservative daughter strands. Each resulting DNA molecule is made up of 1 parent strand and 1 daughter strand. In comparing DNA versus RNA, we have to recognize that DNA is first double-stranded while RNA is single-stranded. DNA contains adenine, thymine, guanine, and cytosine, while RNA contains adenine, uracil in place of thymine, guanine, and cytosine. DNA contains deoxyribose as sugar, while RNA contains just ribose. DNA is nucleus bound, while RNA is in the nucleus and in the cytoplasm. DNA is relatively long, while RNA is relatively short. DNA is stable because of the multiple hydrogen bonds, but also accessible. RNA is not as stable because it's single-stranded, and it has an extra hydroxyl group. There are 3 different types of RNA: mRNA, tRNA, and rRNA. mRNA is messenger RNA, and it carries the code necessary to manufacture proteins. It transcribes the genetic code from DNA into a form from which it can be utilized to make proteins. It is found in the cytoplasm of a cell and manufactured in the nucleus. tRNA is transfer RNA. It delivers amino acids one at a time to the ribosome. The amino acids can then be processed to make proteins and polypeptide chains. It is also found in the cytoplasm of the cell. rRNA is ribosomal RNA. It is found in the cytoplasm of a cell, and it is a component of ribosomes. It directs the translation of mRNA into proteins. Transcription occurs in the nucleus of a cell. DNA is located in the nucleus and mitochondria, but is mainly nucleus bound, while RNA is located in the nucleus, ribosomes, mitochondria, and cytoplasm. When transcription occurs, a primary RNA molecule is created. The primary RNA molecule contains both introns and exons. Later, splicing occurs, and the introns are removed by restriction enzymes, which forms a mature mRNA molecule with the exons attached. This transcribes DNA into messenger RNA. Introns are removed as they are unnecessary and do not code for proteins. For transcription to occur, DNA needs to be unwound and unzipped. During unwinding, RNA polymerase binds to the DNA strand at a specific sequence of genes called the promoter, then unwinds and unlinks the two strands of DNA. During unzipping, RNA polymerase is used again. It does what DNA polymerase does during DNA replication. It synthesizes a short RNA primer, which is later removed. DNA helicase is also utilized in this process. The initiation of transcription in bacteria begins with the binding of RNA primers to the promoter in DNA. Transcription initiation is more complex in eukaryotes, where a group of proteins called transcription factors mediate the binding of RNA primers and the initiation of transcription. Transcription is done using the coding strand, which is the complementary strand for the protein. The mRNA is therefore complementary to this strand. Elongation is when the RNA polymerase lays down bases complementary to the template strand it is reading. Nucleotides are added to the growing RNA chain. The coding strand of DNA has a nucleotide sequence complementary to the template or non-coding strand. The non-coding strand contains the information for the construction of a protein. Elongation is stopped when a stop codon is read. This signals the termination of transcription. After the codon is added, the mRNA strand is edited so that it is able to enter the cytoplasm. The introns are edited out and the exons are spliced together. A poly-A tail, which is essentially a bunch of extra adenines to regulate the number of proteins synthesized, and a 5' cap, which eases transportation through the cytoplasm, are added before the mRNA exits the nucleus. The genetic code consists of 64 triplets, or codons, of nucleotides. With a few exceptions, each codon codes for one of the 20 amino acids. Codons consist of 3 letters, for example, A, U, G. When codons are put together, they make a polypeptide. The code ends with a stop codon and always starts with the AUG. In the genetic code table, the left-hand column gives the first nucleotide of the codon. The 4 middle columns give the second nucleotide, and the last column gives the third nucleotide. RNA codons occur in mRNA. tRNA are floating molecules that are attached to an amino acid on one side and anticodons on the other. They pair up with mRNA aided by a ribosome. rRNA forms the ribosomal structure that aids in building the protein from the amino acids that the tRNA attaches to on the mRNA. It has 2 subunits and 3 binding sites called the A, P, and E sites. We must consider the role of codons and anticodons in translation. Each set of 3 bases on the mRNA is called a codon, and each codon calls for a specific amino acid to be brought to the building site. The first codon is always AUG, the start codon. In the cytoplasm, there are molecules of tRNA. On one end of the tRNA molecule is the anticodon, a set of 3 bases that will match with a certain codon. The other end of the tRNA picks up and holds a certain amino acid. Each tRNA only holds one type of amino acid. A tRNA with an anticodon that matches whichever codon is in place on the ribosome will click into place. The codon and anticodon fit together like 2 complementary pieces of a jigsaw puzzle. Since the first codon is AUG, the first anticodon must have been the matching bases UAC. The process moves along to the next codon. The amino acid brought by the first tRNA bonds to the amino acid brought by the second tRNA. The first tRNA is free to go, and the second tRNA stays in place. The process is repeated. Eventually, the stop codon is reached. At this point, the amino acid chain breaks off, folds up, and the protein is ready to be utilized. An initiation complex for translation forms by the assembly of the ribosomal subunits and initiator tRNA, MET tRNA, at the start of the codon of the mRNA. The ribosome is made of rRNA. The ribosome has 2 parts, which bind on either side of the mRNA. Within the large part are 2 rooms, P and A site, that will fit 2 adjacent codons of the mRNA, 2 tRNA molecules, and 2 amino acids. P site holds the first codon in the mRNA, and the A site holds the next codon. The P site, called a peptidal site, binds to the tRNA holding the growing polypeptide chain of amino acids. The A site, or acceptor site, binds to the amino acid tRNA, which holds the new amino acid to be added to the polypeptide chain. Each tRNA has a binding site for the amino acid. Because each tRNA is specific for a single amino acid, it must be able to recognize the codon on the mRNA that codes for that particular amino acid. Therefore, each tRNA has a specific anticodon that matches up with the appropriate mRNA codon. This is the role of tRNA in elongation of the polypeptide. The process continues in an assembly line fashion, with the polypeptide chain elongating until a stop or terminating codon enters the A site. At this point, the translation will stop, and the completed polypeptide chain will be disconnected to be folded into a complete protein. We do need to address how a peptide bond is formed with amino acids. A peptide bond forms between the carboxyl group of one amino acid and the amino group of the adjacent amino acid. The bond occurs when the carboxylic group of one molecule reacts with the amino group of the other molecule, linking the two molecules together and releasing a water molecule. Long-chain polypeptides can be formed by linking many amino acids to each other via peptide bonds. Substitution or point mutation occurs at a single point. It is a substitution of one base for another. It can potentially mess up overall function and decrease effectiveness, but it's not very detrimental as only one base is affected. Frameshift mutation occurs when a base is added or removed, creating a shift in the reading frame. It results in extremely different amino acids. Adding it at the end would be better than adding it at the beginning, as fewer amino acids would be affected. However, frameshift mutation overall is very detrimental. Inversion mutations occur when a sequence is flipped. The impact of this mutation depends on how large the inversion is. The larger the inversion is, the worse the mutation becomes. A silent mutation is any mutation where the result does not affect the overall meaning. The codon is still affected, but the definition is still the same, since the codon still codes for the same amino acid. A nonsense mutation occurs when a substitution or deletion occurs that creates an unintended stop codon. The impact of this can be devastating, since much of the protein could be left unsynthesized. As a result of mutations, many harmful effects can occur. Silent mutations do not impact the overall meaning, but if point mutation, however, occurs, the polypeptide coded could be altered. In this example, the 7th amino acid being coded for, tyrokinases and phosphatases, is mutated into UUU, which instead codes for phenylalanine. The change in polypeptides impacts the structure, and therefore the function of this protein. This is similar for other mutations, with the exception of the silent mutations as well.